Hi, everybody. My name is Vaidehi Natu. I study human brain development, and today I'm going to tell you about some of the macro and the micro level changes that take place in the brain during early days of human life. So there are several reasons why human development is fascinating. Compared to many animal brains, human brains are less developed at birth and take a long time to mature. So our developmental curve is relatively shallow compared to other primate species. Furthermore, development is a function of several factors, such as genetics, our social and physical environments, and countless experiences with which we adapt to our unique environments. For instance, the visual system, shown here in the schematic on the right, houses several lower and higher order functions necessary for us to perceive and recognize the visual world. Several of these lower and higher order functions undergo prolonged development during childhood. So functions like face recognition that is critical for social communication becomes more specialized for function and behavior as we gain experiences with faces throughout our lives. Similarly, word recognition, another important visual function required for reading and comprehension, undergoes extensive micro and macro level changes during childhood, and this maturation continues through early adolescence. However, the foundational cortical architecture necessary to support these complex functions later on in life is laid down early during infancy and during some of the critical periods of infant development. So in this talk, we will look at some of these microstructural and mechanistic changes that take place during infancy along the visual system's hierarchy. So I wanted to start with this image. So this is a brain image of a newborn baby in our study. And what you see here is that already at birth, our brain has this incredibly folded structure. So you can clearly see the gyri and sulci are in place as cortical folding and gyrification begins in utero. Now, this is the same baby at 3 months of age and then at 6 months of age. And you can clearly see how much bigger the brain got in the first 6 months of life. In fact, there are several large-scale changes that occur in the human brain during this time of development, such as increase in cortical thickness, increase in white and gray matter volume, etc. So there are a large amount of macro-level changes in the brain, but what about micro-level changes? So how do microstructural properties develop during infancy? Our current understanding of microstructural development in cortex is limited as it is based on histological investigations of a handful of sensory motor and prefrontal regions across species. Additionally, imaging research is mainly focused on white matter development of infants. According to histological work, there are two overarching and competing hypotheses about the mechanisms that might underlie cortical development. The first one is cortical pruning that posits that pruning of synapses and unwanted connections might shape our functional abilities. The second hypothesis is related to tissue proliferation or tissue growth, which comes from studies such as this one here shown by Miller et al. So each of these panels here is a myelin stain from infancy to adulthood, and you can see that in both white and gray matter, there's an increase in myelin or tissue growth. And this tissue growth might affect developmental and developmental functional abilities. Additionally, the sequence and the rate of development in different systems also vary with age. So if we focus on this red rectangle here, you can see that the sensory systems develop faster than the higher cognitive functions early in life. And the developmental rate reduces, and then the higher cognitive functions undergo more prolonged development throughout childhood. And studying the microstructural changes in each system will shed light on how these changes will sculpt our neural hardware and behavior. To that extent, we asked 3 questions. First, how do microstructural mechanisms within sensory motor cortex develop during infancy? Is there development in microstructural tissue properties across ventral and dorsal visual streams? And what is the rate and sequence of developmental mechanisms within sensory motor cortex? Typically in our work, we use MRI machines to study development. And of course, we know that using in vivo measurements like T1 weighted scans, we cannot measure microstructural changes. However, we now have in vivo tools to quantitatively measure tissue properties in cortex. And one such method is the quantitative MRI or qMRI, which provides T1 relaxation time, which is inversely proportional to the macromolecular tissue volume, or R1, which is 1 over T1, which is directly proportional to tissue volume. So you can see here in this figure by Metzer et al., and they measured T1 relaxation time. And you can see that in the ventricle, which has almost no macromolecular tissue or is filled with fluid, has a really long relaxation time. But white matter, which is really dense, with tissue has really short T1 or high R1, that is relaxation rate. In fact, tissue with lipids and cholesterol, like myelin, have lower T1 or higher R1. Furthermore, as these quantitative metrics are measured in units and not arbitrary values, as in the case of T1 images, we can actually use them to compare values or compare metrics across brain regions as well as age. So for the sake of this study, I'll present my results in R1, and we have now successfully used this method to study childhood development and validated these metrics, the in vivo MR metrics, with ex vivo histological myelin stains to show that myelin explains R1-related changes in functionally developing areas of the visual cortex. And so we can use this metrics to study if tissue development or tissue growth or pruning happens during infant development. So in this example, I'm showing you a histological myelin-stained slice of the human fusiform gyrus, right here. Darker colors indicate more myelin. And as you can see here, myelin increases from the peel surface to the white matter surface as you move from left to right. Okay, so how does this relate to MR metrics? Here we see a high-resolution quantitative R1 measurement. This is taken at a resolution of 200 microns, and this is of the same tissue that we just saw here in this panel. And this shows that R1 mirrors the myelin content. So again, R1 systematically increases from the peel surface to the white matter surface. Finally, we see the same trend even at 1 millimeter resolution in vivo R1 measurement of the fusiform gyrus. So you can see that there's a systematic increase in R1 from peel surface to white matter. Hence, we can use this metric to assess development of brain microstructure during infancy. So given that quantitative R1 is directly related to microstructural tissue volume, we can make two developmental predictions based on histological work. So first, if development is associated with tissue loss or cortical pruning, then we predict that R1 will decrease with age. Second, if development is associated with tissue growth or proliferation, we predict that R1 will increase with age. Okay, so we conducted a longitudinal study. So in this case, each baby was scanned multiple times. Specifically, we collected longitudinal measures in infants across 3 time points, that is at birth, at 3 months, and at 6 months of age, while infants were asleep inside the scanner. Scanning babies is a little less trivial compared to scanning children or adults. Of course, you can't tell a baby to just stay still or close their eyes. So we first had to create an environment where the baby is asleep during the scanning period. So babies were straddled, and again, babies do get startled and they wake up, and sometimes they wake up due to calibration scans because these calibration scans tend to be a little loud. So before the baby goes inside the scanner, they're swaddled, they're made comfortable, they're put to sleep by their parents, and of course we have a researcher inside the scanner with the baby at all times. And we also monitor the babies during the scan from outside using an infrared camera. And luckily, we've had a wonderful team of researchers, and we also have a really wonderful group of parents and babies. And today I'm going to show you data from our first study with 30 sessions, but we've now successfully collected high quality data from about 100 sessions across 50 babies with a 90% success rate. So from these scans, we collect quantitative MRI, which gives us R1 relaxation time, and T1 and T2 weighted 3D brain anatomies. So once we get the T1 weighted and T2 weighted image, we want to generate cortical surface reconstructions for visualization and for further analyses. However, there are several steps that are involved before we can actually get to these really beautiful surfaces, as baby brains need a lot of cleaning and careful reconstruction of segmentations, as they have really low contrasts between gray and white matter in the T1 and T2 properties, brain images. So once we get the T1 weighted and T2 weighted images, we do several steps to generate the cortical surface reconstructions for visualization and further analyses. So the first step is the generation of gray and white matter segmentation from the T1 weighted image, and we use Infant Free Surfer's automatic segmentation code that was developed specifically for infant data processing. This was developed by Zolay et al. In 2020. The initial segmentation generates peel and white matter surfaces and surfaces of curvature, thickness, and surface area. However, it sometimes misses portions of the infant's gray matter as the content of the T1 weighted images are not differentiated enough between gray and white matter. So to generate more accurate white and gray matter segmentations, we use the T2 weighted anatomical image, which provide a better contrast between gray and white matter in infants. We additionally use an independent brain extraction and analysis tool called iBeat, which was developed by Li Wang's lab. We then manually correct to fix segmentations for any additional errors, such as holes and handles, using ITK gray. And then these manually corrected segmentations are aligned to the T1 weighted anatomy. And the aligned and segmentation volume is further reinstalled in FreeSurfer using an in-house software and FreeSurfer functions. And this process finally updates the white matter segmentation and cortical surfaces to render accurate surfaces for visualization and cortex-based alignment with cortical atlases. So since our goal is to study the changes in R1 or microstructural development in primary sensory areas of visual cortex, we first need to delineate these brain regions in individual babies. So for this purpose, we used cortex-based alignment, and we capitalized on the feasibility in delineating finer cortical maps across subjects via independent adult atlases using cortex-based alignment and projecting these atlases on each individual infant brain. And we used two atlases. We used the Glasser atlas, and we use the Wang atlas in this case. Okay, so to study the development of sensory motor areas, for instance, we used an independent adult atlas, as I mentioned before, and this was the Glasser atlas. And so using this atlas, we draw 4 primary areas on each infant brain. So you can see here that we draw V1, A1, M1, and S1, which are the primary vision, auditory, motion, and somatosensory areas on each individual infant brain. And then after this entire process, this is sort of what it looks like. So we obtained the cortical maps. Then we use cortex-based alignments to project the primary sensory areas on each individual brain, like so. And then we obtain mean R1 values from each of these individual ROIs and individual babies. Okay, so we're going to first look at the question, how do microstructural mechanisms within sensory motor cortex develop during infancy? So I'm going to show you the results now. So here is the cortical surface showing the distribution of quantitative R1 in a newborn baby. So you can see that most of the brain, most of the cortex here is red in color, which indicates low R1. However, at 3 months and by 6 months of age, we see a sharp change in color from red to purple on the cortical surface, indicating R1 is increasing with age in the first 6 months of life. Okay, and these results are very consistent across babies. So these are different babies at the 3 time points that we have. Okay, so now if we look at individual primary sensory areas, so first let's look at V1, which is our primary visual cortex. We fitted a linear mix model to the mean R1, and so we have y-axis on R1. We have on y-axis we have R1, and on x-axis we have age in days, and each dot here represents a baby. So results show a sharp increase in R1 in the first 6 months of life. You can see this really sharp increase right here. What about the rest of the primary areas? And you can see really across all of the regions, we find a consistent increase in R1, indicating significant tissue growth. And so this is, again, very interesting because overall, our prior histological work suggests that maybe our primary areas are actually developed at birth, but turns out they continue to develop in the first 6 months of life. Okay, so now we wanted to examine whether there's development in microstructural tissue properties across the ventral and dorsal visual hierarchies. So within the visual system, again, we examined the development in the ventral visual system first. And here we used, again, cortex-based alignment and the adult Wang atlas to project the individual brain areas in the ventral visual stream onto each infant brain. So you can see here from the posterior to the anterior, we're going to look at how R1 changes across each of these different areas in the ventral visual cortex. So first, coming to the results again now, I'm going to first show you results from the primary visual area V1. So here again, on the y-axis is R1 and x-axis is age in days, and the dots represent individual subjects. And we fitted a linear mixed model again to the data. And what you can see here is that in V1, just as we saw before, there's a sharp increase in R1 across age. So what about the rest of the areas in the ventral temporal cortex? And you can see that pretty much across all areas, there's an increase in R1 in the first 6 months of life. Okay, so what we did next was we were also interested in looking at two parameters. The first, we asked if the distribution of R1 at birth, represented by the intercept of these linear models, is uniform or not. And second, we asked if the rate of development of the regions, represented by the slope of these linear mixed models, varied across RYs. So this is R1 at birth on the y-axis, and x-axis is each region along the ventral visual stream. And what we find is that there's a systematic decrease in R1 at birth, ascending the visual hierarchy of the visual system, or the ventral visual processing stream. So the primary visual area has a much larger R1 at birth, suggesting more tissue compared to the higher order visual areas. So what about the rate of development? So here again on the y-axis is the rate of development, and the areas are on the x-axis, and larger this number, faster the rate. And we found that the changes in tissue properties in the first 6 months of life become progressively increasing from the primary visual areas to higher areas in the ventral visual stream. Okay, so what does this mean? First, our overall results suggest that there's extensive tissue growth in the visual cortex. However, development is heterogeneous and hierarchical, as early visual areas are denser at birth, but develop at slower rates than higher order visual areas. So this suggests that the cortex is not actually, in fact, uniform at birth, but there's a hierarchy. And perhaps V1, for instance, is more developed at birth to be ready to take in the input from LGN to get the visual system going, and the rest of the areas then come online following birth. Okay. And what is interesting is that we again find very similar developmental pattern in the dorsal visual stream, which we know is involved in visual attention and the Ware pathway. So this is the rate, this is the tissue density at birth, and this is the rate of development after birth. Okay, so now that we see tissue growth during infancy, we asked what brain mechanisms might be developing in the visual cortex during early infancy. And this is, again, a very important question, because if you were in the white matter of the brain, where you see a lot of myelin-related changes, of course, then we would think that this growth that we see in R1 might be related to myelin, right? However, this is cortex. And so in cortex, again, it's interesting because there's many other things that could be contributing to this tissue growth. So to address this question, we made use of this openly available transcriptomic gene database, and this is the BrainSpan Atlas. And the idea here is that thousands of genes are expressed during each stage of human development and in different parts of the cortex. And the expression levels of the genes varies depending on age and the environmental factors. So if we were looking at the same brain regions that we tested in our babies, we can ask what genes are most differentially expressed during the postnatal versus prenatal stages of human life in this tissue samples, and what are the cellular mechanisms that might be associated with these individual genes. So this is what the BrainSpan Atlas page looks like. Again, this is a very rich data set that allows differential gene analyses. And we can do this sort of gene analyses across multiple areas, right? You can see all these different areas of the brain, and also across, a really range of ages. So you can see that these samples really range from gestational ages all the way to adulthood. So it's a very, very rich genetic database. And so using this database and this analyses, we can generate, these beautiful heat maps. And this is a heat map representing gene expression levels. So in this case, purple represents high expression levels and green represents low expression levels. So each column in this case is a tissue sample. You can see here, this is a tissue sample from the brain areas in either postnatal, so this is postnatal, or prenatal stages of human life. And each row, in this case, each row right here, is a gene arranged in descending order. So the big take-home message from this analysis is that the top most differentially expressed gene in our analyses turns out that are predominantly related to myelin. So you can see that the topmost gene, in fact, is myelin basic protein, which is associated with myelin generation. Then we have myelin-associated oligodendrocytic basic protein, proteolipid protein, and myelin-associated glycoprotein. So a lot of these genes are associated with myelin and myelin generation. So to further elucidate which specific molecular and cellular pathway might be linked to our top genes, we used the top gene toolbox. So this is a very nice tool. Again, this is a tool that allows us to map this list of expressed genes to the enriched physiological processes. So this is what the top gene suite looks like. And so for our analyses, the top gene toolbox reported significant enrichment of several biological processes related to the genes. In fact, some of these processes are listed here. So again, typically what our results suggested is that the transcriptomic data reveals that myelination and synaptic processes are two dominant mechanisms underlying the tissue growth in visual cortex during infancy. So you can see a lot of these processes here are associated with myelin. So again, there's structural constituency of myelin, there's myelination, there's axon ensheathment, and then there are other processes that are related to synaptic processes, right? So again, these are sort of the two predominant or dominant mechanistic processes that we think might be associated with this tissue growth that we see in during infancy. Okay, so to conclude, we think our results showed profound tissue growth in gray matter of the visual system. Again, we think that myelin might be a key factor in the gray matter development during infancy. And this might be a critical piece of the developmental puzzle for the functional improvements that we see later on in childhood. And finally, I just want to highlight the importance of combining multiple measures of microstructure and morphology with postmortem and transcriptomic data to study mechanistic processes underlying human brain development. And last but not least, I want to thank all of my collaborators, especially Dr. Mona Rosenke and Dr. Colony Grill-Spector, our entire VPNL lab at Stanford, our global collaborators, our funding resources, and thank you so much for listening.